Okay, everyone, we're back in the lab for our blood typing experiment. So, I'm going to run through how you would be doing this if you were in the lab, but make sure you pay attention because I'm going to be asking you questions about this on our lab quiz, and then also I'm going to be asking you questions about the process and the results on your lab assignment. Are you ready? Okay, so what we're going to do is we have 4 fake blood samples. So you can see here this says, can you see that? Yeah, Mr. Smith. It's not real blood, it's fake blood. But this process is very similar to what they would do for real blood. Excuse me. So we have 4 different fake blood samples, and I've labeled them out right here, okay? So we can see we have a Mr. Smith, a Mr. Jones, a Mr. Green, and a Ms. Brown. It's a veritable clue of biology laboratory. I'm excited. So what we're going to do is we're going to follow the instructions that would be in the lab, and the instructions say to put 3 or 4 drops of each fake blood sample into each of these wells. I've labeled them so you can see. On each little plastic well, there's an A, a B, and an RH, because these are the antigens that we're testing for. Remember? A person can have either A or B antigens on their red blood cell surface. Remember, if they have an A antigen on their red blood surface, then their blood type is A. If they have the B antigen on their red blood cell surface, then their blood type is B. If they have both the A and the B, then their blood type is AB. And if they have neither, their blood type is O. Okay? Then we're also going to have a separate series of antigens called the RH antigens. If you have the RH antigens, then your blood type is positive. If you don't have the RH antigens, then your blood type is negative. So let's say that someone has the A antigen and the RH antigen. Their blood type would be A positive. If they have only the B antigen but not the RH, they would be B negative. So here's how we set it up. And in the lab, I always tell people, if they can, put it on a white sheet of paper because it's going to be easier for them to see the results. I'm going to grab a stool. Turns out it's hard for me to bend down that much. It's like a lab wall sit. Okay. So here I have Mr. Smith, and I'm going to put 3 drops in the Mr. Smith section. So again, this is fake blood. If it were real blood, I'd be wearing gloves. So I'm going to put 3 drops of Mr. Smith in the Mr. Smith A. 1, 2, 3. RH. 1, 2, 3. And the B. 1, 2, 3. I'll do the same thing for our 4 other samples. Mr. Jones, and I've labeled it over here, Mr. Jones. 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay. Mr. Green. 1, 2, 3, 1, 2, 3, 1, 2, 3. And then lastly, we have Ms. Smith. Apparently she's not married. Ms. Smith. And we're going to put 1, 2, 3, 1, 2, 3, 1, 2, 3. Boom. And I'll bring the camera closer to these when we actually get to the part that's really important. All right. Next, we have, and I apologize, they seem to be doing work in the lab. We have an anti-serum. We have an anti-RH. We have an anti-B. And we have an anti-A. So here's what I'm going to do. The anti-A. So we're basically adding the antibodies on purpose. In real life, we wouldn't want to do this. We wouldn't want to add an antibody that might possibly bind to a matching antigen and then clump with it, agglutinate, and blow up the cell because you would die. But in this experiment, this is going to be the way that we figure out which antigens are present. So I'm going to take the anti-A, and into each of the A's, so an A well here, A well here, A and A, So I'm going to add the anti-A, or the A antibody, to the A's of all four. 1, 2, 3. 1, 2, 3. 1, 2, 3. And 1, 2, 3. And you guessed it, we're going to add the anti-B to all of the B wells. As I'm doing this, we can certainly review. So we just talked about whatever antigen is present on the red blood cell surface. That's what blood type you have. So what if you have no antigens and your blood type is O? And O is our universal donor. So O is a really valuable blood type. Oftentimes we can't mix the blood types. So if I'm blood type A positive, if I need blood, I'm getting in some horrible accident and I lose all this blood and I'm in the hospital. If I need blood, they've got to give me A blood type because I'm A blood type. But they can also give me O blood type because O is the universal donor. And the way that I remember that is donor has an O in it, donor, D-O, and then O is the universal donor. So we have our anti-RH antibody, so I'm going to add that to each of the RH wells. More review. So if O is our universal donor, type AB blood is our universal recipient. So if you out there are type AB blood, that means that you can receive blood from anybody. So AB is a universal recipient. You should know those. All right, great. So now that I've done this, we're going to take a toothpick and we're going to mix them up. So remember, we have the antigens added, the A antigen in the A well, the B in the B well, or excuse me, we have the blood type in each one, and then we're adding the antibody. What we're going to be looking for is for the antibody to find its matching antigen and what's called a glutenate or clump. It's just like it is with the immune system. An antibody is able to attach and bind to and break down a matching antigen. So let's say we have the chickenpox antibody. Well, the chickenpox antibody can bind to the chickenpox, break it down and kill it. So here, the anti-A antibody is going to be able to bind to the A antigen and break it down. Again, we wouldn't want to do this in real life, but outside of the body, it's our way that we can determine what antigens are present in the blood sample. So we're looking for that clumping or agglutination. So when you see agglutination, and I'm going to show you, I'm going to mix up our samples, and if we see little clumps or little dots, then we're going to say it's agglutinated. If we don't see any dots or clumps, then it is not agglutinated. Now, sometimes it can be a little bit hard because it may be cloudy, but just because the solution is cloudy does not mean that it has those clumps, okay? So usually when it clumps, it also turns cloudy, but I just want to make a point that cloudiness alone is not what you're looking for. We're going to be looking very closely for little clumps, and that's going to be positive. Okay, so I'm going to pull out my clean toothpick, and I'm going to mix it here. How can I do this? I might have to move my face, not that you're sad about that. All right. Okay, high-tech science right here. So I'm starting with Mr. Smith, and as I mix it up, I don't see any clumping. Oh, wait a minute. As I move it around more, I do see tiny little clumps. It's really unfortunate that through this video you can't tell. What I'm going to do is I'm going to post an additional little video showing the clumping up close, but I wanted to go through this in person. And let me grab my marker real quick. Hold that thought. I'm back. That was a dramatic pause, right? So I'm just going to put a little mark, okay? So the A clumped. So if I circle it, it means that the A clumped. So we do see little dots there. So it clumped, it agglutinated. I'm going to switch sides and use a clean side to do the B well. And in the lab, you can see that we're going to do this one at a time. And I'm going to do this for a while to see if I can really make sure if it's clumping or not. And what do I see here? No clump. Get a fresh toothpick. And now I'm going to do the RH. Oh, didn't happen at first, ladies and gentlemen, but through the magic. Oh, yes. So, in this first example, the A and the RH clumped. Excuse me. How you determine the results, whatever clumped, it is that blood type. So if the A clumped, I know Mr. Smith has A blood, and then I look to the RH. If it did clump, it's RH positive. If it hadn't have clumped, it would be RH negative. But since the A and the RH agglutinated or clumped, the blood type is A positive. Now for the rest, I'm going to mix it up, but I'm not going to tell you the answers. That's going to be for you to decide. So clean side, let's go down to Mr. Green. Okay, I can already see Mr. Green here. The A has definitely clumped. Let me get a fresh one. Let's look at the B. At first, it didn't look like it was clumping, but as I'm mixing it up, it's clumping. So A and B has clumped, and then we'll do the RH, and the RH is ridiculously clumpy. Mr. Green is a clumpy man. So in this case, we have the A clump, the B agglutinate or clump, and we have the RH. So I'll let you determine what blood type that is. Let's continue on. Let's do Mr. Jones. I'm going to do the A. Nothing. I'm going to do the B. Oh yes, we got some little dots moving around in there, some little chunks. So far for Mr. Jones, the B has agglutinated. And let's check the RH to determine whether that B goes along with an RH positive or an RH negative. No clumping. I got nothing. So for Mr. Jones, it was only the B that agglutinated. Last one, Ms. Brown. Let's do the A. Nothing. Last little toothpick here. Let's try the B. Nothing. Fresh side. And let's try the RH. Yes. So in Ms. Brown's case, we have, you can see all of them. Ms. Brown's case, only the RH clumped. So I'm going to let you determine what blood type that is. So folks, that's how they do the blood typing. I'll let you get the rest of the results. Good luck.